Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon, I'd like to welcome you to Targeted Approach to Metabolomics. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. At this time, I'd like to introduce our three presenters for today. First is Robin Parham, Targeted Analysis from Metabolon. Our second presenter is Richard Robinson, Staff Scientist at Metabolon. And our third presenter today is Dr. Brian Kepler, Associate Director of Metabolon. So welcome, Robin, Richard, and Brian. The presenter ball is yours.
Okay, hi everyone. Yeah, I'm Robin Parham. I'm the project manager of our targeted analysis team here at Metabolon. And I'm going to talk to you today about our targeted approach to metabolomics. So first I'm going to start with talking to you about what is targeted analysis here at Metabolon. Move into some of the features and challenges that we deal with on a day-to-day basis, and then talk to you a little bit about how we might design an assay and how a project might work from beginning to end. Tell you what our focus panel menu is currently, and then talk to you a little bit about a couple studies that we've worked on, collaborations that we've done with some different clients. So first, what is targeted analysis here at Metabolon? We do basic quantitative biomarker analysis. So we're dealing with small molecular endogenous metabolites. We use mostly LC-MS instruments. We do have one GC-MS that we use for our fatty acid panel. We also support our R&D group doing biomarker isolation and identification. And we also have a synthetic chemist on staff in our group who can synthesize reference standards and stable label and turtle standards to make sure our assays are very robust. So how do we kind of work in with how Metabolon works here? So we have our biomarker discovery platform that I'm sure many of you are familiar with, screening for many, many metabolites. That data is then mined by our biochemists and statisticians, and at that point it comes to us in the targeted analysis group. So we have the ability in our group to take a small volume of plasma, urine, feces, whatever the case may be, and run it through an extraction, and treat that extraction several different ways. And in this case, we're monitoring for 25 analytes over 4 different methods on many different instruments. At that point, we are able to run it back again through our biochemists and statisticians, narrow those analytes down even more to eventually go into a production assay, possibly in our CLIA group, diagnostic test, bioreactor monitoring, whatever the case may be. So how are we in targeted analysis different than our biomarker discovery platform? On the discovery platform, they're doing relative quantitation. So you're screening for thousands and thousands of metabolites, and the changes in a given sample are analyzed relative to, say, an untreated control sample. This is really a great screening tool for early biomarker discovery, and you can we also can run metabolic disease screening, such as our Meta-IMD. In targeted analysis, we're doing absolute quantitation. So at that point, you're targeting in on anywhere from 1 to, say, 40 metabolites. We use a standard curve method, so the unknowns are quantified based on a known quantity that's spiked into our standard curve. And we provide accurate quantitative measurements. So for, say, biomarker data and drug development studies, we can do preclinical or clinical and even diagnostic tests. So I'm sure everyone's familiar here with how accuracy and precision work, but this just goes a little step above and say that in the targeted assay group, we're doing absolute quantitation. So we really need to hit that concentration on the target and hit it consistently. On the platform side, we're looking at relative quantitation. So there really is no target. There really is no true concentration. We're really just looking at precision at that point. So some of the technical challenges that we deal with when we're developing an assay, one of the biggest ones is multiplexing these large number of analytes to hone down in on just a few assays. We also are dealing with really trace analysis, so very sensitive compounds we're looking for. So just to put that into perspective, 1 nanogram per mil is an average concentration that we're looking for in many of our studies. So if you think about that, you can take 12.5 ibuprofen tablets, 200 milligrams each, and dissolve that into an Olympic-sized swimming pool. And that's 1 nanogram per mil. So we're really trace analysis here. On top of that, we're not looking at water. We're looking at plasma or feces or urine. So, we really need to make sure these samples are cleaned up and injected on the instrument appropriately. In addition, we're dealing with these endogenous metabolites, so isomers and interferences are always going to pop up, so we really need to make sure our methods are very selective. And we know that clients' samples are precious, and so we want to minimize the amount of sample that we use in an assay, so we want to make sure we're dealing with really small sample sizes. So how would a project work if you were coming to the target analysis group? We would start with a sponsor-led consult, so everybody would get on the phone together, find out what you're interested in, what the needs are, what the turnaround times are, and then move into a scientific assessment and quote. So you would be getting a free scientific consultation, basically, from one of our staff scientists, and determine whether you're going to be running a custom assay or a pre-developed assay. If it's a custom assay that you're requiring, we would determine that multiplexing capabilities. We would determine whether the standards are available commercially, and if not, we can synthesize them from our synthetic chemist. And if you're looking at a pre-developed assay, you would choose from the menu that we have available, and I'll go through that soon. Finally, pricing would be provided in a quote, and then we would move into project execution. So per that contract, we would synthesize reference standards as needed. We develop the method, we validate it if it was a custom assay, and then analyze the samples for you. And the final deliverable for a validation, method development and validation is a report, method validation report, and then for sample analysis, a sample analysis report. So for a custom assay, what are we going to be doing? In method development, we are going to source those reference materials or synthesize as needed, optimize our extraction method, optimize our mass spec conditions, develop the chromatography, and also run range-finding samples to determine what the appropriate calibration ranges are to make sure that our samples fall within that range or can be diluted within as needed. Once that method is developed, we would validate it according to the regulatory requirements that are needed by the client. We can run our research use only studies or good clinical practice studies as well. Then we would take that validated method and run your samples. Your final deliverable there is a sample analysis report, which would include a brief description of the method, how the method performed with some QC data, and then obviously your data. So we have a couple different levels of validation that we have available here. The lowest level is kind of rapid data, so just kind of an informal qualification of the accuracy and the precision that we would kind of run during method development. So this is pretty informal and more of just a kind of a quick look kind of method. Next one up is our method adaptation, which would more be if we already have the assay validated in maybe plasma, switching to feces, or if we're switching the species of the assay, that would just be a one-run validation. The most typical that we run here is the full research use only validation, which is a 3-run validation, following as closely as possible the FDA guidelines without going to our top tier, which is GCP, where we run in full accordance with the FDA guidelines. We include our QA department. We have tons of documentation for that, and that's a 6-run validation. So here's our list of our pre-developed focus panels that we have available here at Metabolon in the targeted analysis group. We have a bile acid panel. We have an amino acid panel, a free fatty acid panel, total fatty acid, which is the fatty acid methyl ester panel. We have short-chain fatty acids available, sterols, which is cholesterol metabolism panel. We have a Sebum lipid panel that Richard is going to be talking to you about shortly. Stratum corneum lipid panel. We have accuGFR available, which Brian will be talking about soon, and also a metal ion panel. We also have existing custom developed assays that we've done over the years. At this point, we cover over 300 metabolites that we have worked on in various matrices, biofluids, tissues, human, animal, whatever the case may be, coverage across really the whole metabolome at this point. Occasionally, you can use these as is or with limited adaptation, again, if you're trying to switch from one species to another or different matrices. So now I'm going to just run through a couple different studies that we've collaborated with other clients, just to give you an idea of kind of some of the studies that we've worked on and how those kind of ran. So this first one was a large pharmaceutical company. They were developing a drug for the treatment of NASH. It was an FXR receptor agonist. They found that C4 and bile acids were PD markers for FXR agonist activity in vivo. So we developed an assay for them for C4 and 15 bile acids, and the bile acids are a panel for us. And we analyzed human serum and feces for them. This was a very large clinical study, over 12,000 samples that ran over about 6 months. The samples were sent to us batch-wise, and we had a 2-week turnaround time between the time the samples came in and the data went out. And this was required for adjustment of dosing levels for the next cohort. So it was a really nice study, ran very smoothly, and it was a really fun one to work on. The next one here is actually also another large pharmaceutical company, also developing drugs for the treatment of NASH. This one was a fatty acid biosynthesis inhibitor. We started with our biomarker discovery platform with this one. So really, kind of almost beginning to end, identified 2 compounds of interest. Malonyl carnitine was found as a PD marker for FASN, fatty acid synthase inhibitor activity, and malonate was found as a PD marker for ACC inhibitor activity. We developed assays for these 2 analytes, and we analyzed samples, and this is kind of currently an ongoing study as well. So, next one to show you is a medium pharmaceutical company we worked with treating a rare genetic disorder, long-chain fatty acid oxidation disorders. They were tracing the metabolism of nutritional short-chain fatty acid product as an alternative energy source to long-chain fatty acids. We developed an assay for 3 endogenous metabolites in human plasma, and this is actually a GCP study, and again, is ongoing. We've had multiple clinical studies with them running from phase 1, 2, and 3, so it's been exciting to kind of see this one come to fruition as well. Next one is a small pharmaceutical company. We helped with metabolic characterization of intestinal bacteria strains for them. Short-chain fatty acids are produced by the gut microflora, beneficial for maintaining gut health. They were dealing with a microbial culture-based drugs for the treatment of gastrointestinal diseases, for example, after antibiotic treatment has been done. And we helped them in characterizing their strains by measuring the short-chain fatty acids for them. Finally, last one was pretty exciting. This is a large food and nutrition company, might be able to figure out. They were dealing with a new infant formula. There was a formula with prebiotic human milk, adding the 2-fucosyl-lactose. So they were looking at comparing breast milk fed infants with infants fed with formula containing this fucosyl-lactose, which wasn't previously included in most formulas. So we developed the assay and analyzed study samples for this 2-fucosyl-lactose in infant plasma and urine. So this was a really exciting one to kind of see come to final fruition involving a product on the market now. So why come to us? Why come to us for targeted assays here at Metabolon? We really can provide beginning to end, starting with metabolomics discovery, all the way down to a diagnostic assay. We specialize in complex multiplexed assays, and we have the capability to synthesize these reference standards using our synthetic chemists. That really sets us apart, we feel. We have a wide range of high-quality assays, whether they're the pre-developed or custom assays to suit whatever the client's needs may be, and have the appropriate quality systems to fit the regulatory needs as well. We have capacity to run a large number of samples and do it fast. And most importantly, I think we have a real consultative approach, routine scientist-to-scientist communication, and really personalized attention for our clients. So that's just kind of the overview of how things work in the targeted analysis group. I'm going to pass it on now to Richard to talk a little bit more about the Sebum panel we have available.
All right, thank you, Robin. As mentioned, I'm going to give sort of a brief overview of how we were able to do comprehensive and quantitative lipidomic analysis in Sebum. So first, let's start with what is Sebum. So if you sort of take your finger and run it down the side of your nose, the waxy, oily substance that you can feel on your fingers there is sebum. And sebum is known to have multiple functions on the body. One of the most important is in helping the skin's role in water retention, which is one of the most important roles the skin has. It keeps the water that's inside you, and the water that's outside you. Otherwise, we would all dissolve when we took a shower. There's also a role played in wound healing. This material is photoprotective and has antimicrobial properties. Analytically speaking, sebum is very interesting because it has a rather unusual lipid composition. In addition to cholesterol esters, di, and triglycerides, sebum has some unusual fatty acids, including branch chains in both the iso and anti-iso forms. And human sebum, at least, contains the rather unusual fatty acid sapienic acid, which is an isomer of the 16-1 fatty acid. The normal isomer is the N7. The N10 is the sapienic acid, which, as you might be able to guess from the name, is found almost exclusively in humans. Furthermore, sebum also contains wax esters, which are an unusual class, and also has a rather large contribution from squalene. So why are people interested in studying sebum? Historically, the interest has largely been from the cosmetics and the skincare industry because of the known role that sebum has in acne and in aging. However, sebum is also thought to be involved in various other skin diseases such as rosacea and psoriasis. And hopefully a better understanding and analysis of sebum can allow understanding of disease mechanisms, allow companies to evaluate therapeutic claims, and allow us to help link skin conditions to diet. Furthermore, there's at least some evidence that we may be able to use the sebum as a non-invasive readout for whole body lipid metabolism. Sebum sampling involves affixing a small sticker, a Sebum tape, to the forehead for 30 minutes. So it's clearly very non-invasive. And then we run into the question of how do we best understand the lipid composition of this material? Unfortunately, historically, a lot of sebum analysis has reported results in very bulk terms, as you can see from the figure on the right of this slide, where results have come back as total triglycerides, total squalene, total lipids. And unfortunately, while that can be sometimes useful and provide insights, such as you can see here, where people with acne have more sebum, that isn't really as useful as it could be. And to understand why, we sort of have to take a step back and look at complex lipids as a whole. And to really understand complex lipids, you have to analyze them very holistically. And the reason for that is that complex lipids they consist of a head group with one or more fatty acid tails attached, and the head group can be anything from a simple glycerol backbone to a cholesterol moiety to, in the case of the wax esters, a fatty alcohol. And what's really important is not to think of these as necessarily individual species, but to try to think of them as a matrix, where one array is essentially the head groups and the other array is the fatty acids to make them up. When you think about it this way, you can start thinking about what's going on across the lipidome with an individual fatty acid, so i.e. looking sort of across one of the rows in this matrix, or what's going on within all of the tags, so looking down the column, or even sort of taking a subgroup of that and looking at what's going on with odd chain fatty acids within this class, or what's going on with monounsaturated or polyunsaturated fatty acids, so that seeing the lipidome as a whole is more important than seeing any individual species. So this does mean that there are certain very important requirements for any analytical method in order to enable that sort of analysis. One, it has to be specific. And when I say that, I mean it has to be specific down to the level of the individual fatty acids that comprise that species. Having some total, i.e. DAG 36.2, isn't really as useful as being able to say this DAG has two 18.1 fatty acids, or an 18.0 and an 18.2, or some other combination. Because without that level of specificity, you can't map that onto this matrix that we're talking about. Secondly, the method has to be quantitative, because without good quantitation, the ability to sum those cells either across rows or down columns or any combination of those things disappears. You also have to have sufficient coverage. If you don't have enough of those cells filled in, then your ability to actually power any sort of study is lost. And for sebum specifically, there's an additional requirement that you need to have a way to deal with those isomeric fatty acids, those iso versus anti-iso, as well as the sapienic acid and other positional isomers. So how do we go about solving all of these problems? So our solution we've dubbed the Sebum Lipid Panel, and it's a two-part solution. And the first part is based on Metabolon's flow-inject MS lipidomic platform that we use for complex lipids. And in that case, the analysis is flow injection based, and it's a series of MRMs. And these MRMs have been very specifically designed to allow us to identify those fatty acids based on the fragmentation pattern. The quantitation part is supplied by the fact that within each of those classes, there is at least one, and for most of them, multiple isotopically labeled internal standards. And these standards are added during the extraction process so that they are able to account both for changes in extraction efficiency and in ionization efficiency to allow us to correctly quantitate all of those species. Additionally, the MRM list not only allows us to identify those fatty acid side chains, but has been specifically targeted to those lipids known to be present in sebum. To address the final question as to how we deal with the isomeric fatty acids, there's actually a specifically designed FAMES panel that is performed in order to correctly identify those fatty acids, which is actually done on the bulk material. So in the process of doing the validation for this assay, we did a study to do some biological validation, and I'd like to just walk through a couple things that we found that were surprising, and a couple of things that we found that just confirmed things we sort of expected to find. So first of all, we actually had 40 subjects, 17 female, 23 male. Actually, these are 39 Metabolon employees and one local television personality who happened to come by our labs filming and saw people with stickers on their foreheads and asked, what in the world's going on here? And when we explained, he said, hey, I'd love to be part of that. So one of those males is actually a local minor celebrity. So what we did find is that consistent with the literature, the males did produce approximately 40% more sebum than females, at which point I will refuse to make any statements about whether or not males just have greasier faces. In addition, what we found, consistent again, was that males have a significantly higher contribution of odd chain fatty acids than do females. But interestingly, and this is one of the things that we could not have seen without that specificity and quantitation that this assay has, is that difference was not evenly distributed among the different classes, but those differences were highlighted within the free fatty acids and the tags, and not seen in the wax esters. And furthermore, what we also noticed, which was a little bit unusual, is that the DAGs actually contained a higher percentage of these odd chain fatty acids than did any of the other classes. So it was very nice to see both things that confirmed what the literature had said, but also to be able to, as you can see here, to add, based on those important properties of this method, the ability to have that quantitation and that specificity to add these new pieces of information. Further, when we looked at the wax ester classes, what we saw was that there was actually quite a difference between the fatty acids and the fatty alcohols in terms of the chain lengths and levels of unsaturation. So what we saw actually is that the fatty alcohols are significantly different distribution than we saw with the fatty acids. So all in all, what we saw is that what we have here is a method that allows us to profile more than 900 lipids in human sebum. Not only that, we can quantify the individual lipid concentrations. We have that specificity that I've talked about several times that allows us to resolve the individual fatty acid compositions within those lipids. In addition, with a 13-minute runtime and a highly reproducible method, we are in fact able to run even relatively large studies over this method. And with that being said, it really only allows me to thank, and I don't have time to actually do so specifically, the numerous people who've been involved in helping to design and run this assay. And with that, I will pass this over to Brian.
Thank you, Richard. So the next part of the talk is going to be about accuGFR, which is a novel test for the precise determination of the glomerular filtration rate. So this is an endeavor undertaken by Metabolon with some collaborators in which we have developed this test which is more precise and accurate than the standard of care estimated GFR values. And I'll take you in the next few slides along the pathway and the progression of which we did this, starting with the discovery platform all the way through targeted analysis. The first thing I wanted to show you is a little bit of background about why we decided to undertake this, this mission to identify better markers for GFR. So the gold standard for GFR is measured GFR. So mGFR is using exogenous filtration markers such as iohexol to determine what the GFR is in a patient. Now instead, because mGFR is more cumbersome for the patient, it's expensive, it's not practical for routine clinical practice. Instead, what is commonly used are endogenous markers such as creatinine or cystatin C. So those are used to estimate GFR, or giving us an eGFR value. And these are used in validated equations such as MDRD or CKD-EPI. The limitations of these, however, is that some of these equations are dependent on some demographical features such as race, gender, and age. The point I wanted to make is that when you plot mGFR versus eGFR MDRD, you see a spread of the data. If everything was equal and ideal, you would see that an mGFR value of 60 would be an eGFR value of 60. However, that is not necessarily the case. This is, again, using endogenous markers, creatinine in this case. Creatinine is found in the muscle and in the diet. So, again, some of these things that are involved in the equation are not under full control like mGFR is. So the limitations here is that eGFR basically there's some leeway for improving accuracy and precision. So the hypothesis that Metabolon has was, okay, creatinine is a metabolite that is filtered by the kidney. Perhaps there's some additional metabolites that are better representative, more accurate, and more precise to estimate the GFR. I just want to give you a little bit of background about accuGFR, what it is, basically in a nutshell. It is indicated as an aid in the diagnosis and management of patients with or at risk of having kidney disease. Some additional information, again, it is independent of creatinine or cystatin C, and thus there are no demographics. So these eGFR equations, eGFR CKD-EPI and eGFR MDRD, these are dependent on demographics such as, again, race, age, and gender. The reasons for that is, again, creatinine is is a metabolite largely found in muscle. So different races inherently have increased muscle mass. Genders, males have more muscle than females in general. Age, when sarcopenia happens in the elderly and you have muscle wasting, those sort of things can affect this algorithm, these standard of cares, which are commonly used up to today. Now, we see an improvement in diagnostic and GFR staging accuracy, also an improvement in CKD clinical trial efficiency with our accuGFR test as compared to the eGFR. So what does accuGFR measure? It is 4 metabolites. They are acetylthreonine, phenylacetylglutamine, pseudouridine, and tryptophan. These are 4 metabolites relatively unrelated to each other. One is a nucleotide degradation marker, one is an amino acid, one is a microbially related metabolite. And really, the importance of having these linked to the kidney is not really relevant. Creatinine itself is not linked to the mechanistic functionality of a kidney. It's just a metabolite that is filtered by the kidney, just as these other 4 metabolites are. It just so happens through our analysis, our discovery, our targeted assays, we have identified that these 4 metabolites are better markers than what is commonly used in those standard of cares with creatinine. So this uses one algorithm, one LC-MS method. It is validated in serum and plasma and clinically validated with the algorithm in serum. And the last thing to note here on this slide is that it's a very robust process. I'll go over the progression and the process in the next few slides. We use large patient cohorts, and this was also done in collaboration with a number of the top key opinion leaders in the area, in this field, Drs. Levy, Inker, and Koresh. These are the KOLs who actually developed the eGFR MDRD and eGFR CKD-EPI equations. So it's important to note that our work is not done in a vacuum. It's not done in competition with these other equations. It's meant to be an improvement upon these methods, and we do have the full support of these KOLs. So here's a look, an overview of the test or the panel product development process. So again, we started with the Discovery Global Metabolomics, which Metabolon offers, and this led to assay development, optimization, analytical validation, and clinical validation. Again, these were using a number of large cohorts, upwards in the high hundreds and thousands. They were relevant cohorts, fit for purpose. They were very large cohorts, so it's giving us confidence in our data, giving the clients confidence as well. And they were also independent cohorts. They were unbiased. Now, the biomarker selection and attrition process, again, started with global metabolomics. So from this process, we measured upwards of 1,000 different metabolites on the global panel. 60 metabolites, and I'll show some of these on the next slide, were found to correlate positively with the GFR values. We then went through assay development, assay optimization, narrowing down the number of metabolites, the number of methods to, again, those 4 metabolites I mentioned, acetylthreonine, pseudouridine, phenylacetylglutamine, and tryptophan. Those 4 analytes are what is comprised in the single algorithm on a single method for accuGFR. All right, so here is some of the data from the biomarker discovery. So the first step, global biomarker discovery, global metabolomics. So the data you see here, there's 3 columns looking at 3 different cohorts. One is the ASK cohort, the African American cohort of hypertension and CKD. The second one is the RIACI cohort of Italians with type 2 diabetes. And the third one is an Irish VHI cohort of healthy individuals. So some of the things to take note of on this data is, one is, regardless of the clinical setting, we do see the same top markers. So the metabolites listed in the first column on the left, you see, and if you see the rank of correlation, it's really the right side of each of the 3 columns. You can see that these are all highly ranked and correlated with mGFR or eGFR. The second thing to note is that this platform does actually rediscover the known clinically useful markers. So that is urea and creatinine highlighted in orange in the table there. So we know urea and creatinine are things filtered by the kidney. They increase in the blood when the kidney is not functioning properly. And these actually show up in our initial screen. And the last thing to note is that these are not actually the top markers. We actually discovered markers with better performance than these currently used markers. The next thing I wanted to show you was some progression in developing various equations along the way and using different models and how we monitored whether or not accuGFR was outperforming eGFR MDRD. So this is a plot. This will give you a good idea of looking at, this is about, I think it's about 1,700 individuals. And mGFR is plotted on the x-axis versus eGFR MDRD on the y-axis. So again, all things being equal, these would all lie on the diagonal. However, eGFR is just an estimation, as is accuGFR, however more accurate. So if you look at the spread of the data, the P30, if you will, I'll talk about this later. The P30 is the percentage of metabolites, percentage of samples within plus or minus 30% of the mGFR. You can see that there is a good spread of the data and that there are some false negatives and false positives highlighted in orange. A figure on the right-hand side here is how the accuGFR at this point, the Model 2 with the equation at this time, actually squeezes down these samples. So you can imagine the spread of the data on the left-hand side, if it were squeezed down and more tight, less false negatives, less false positives. That actually leads to a rate of misdiagnosis at GFR 60 to be decreased 32% versus MDRD. Okay, so some other performance metrics in terms of looking at this data are, one is the P30 that I mentioned before. So the outside dotted lines on the diagonal here, that's the plus or minus 30%. Another thing to look at is the staging concordance. So stages of kidney dysfunction, if your GFR is high, it would be on the right-hand side of the x-axis. It would be healthy. If you're in stage 5 of kidney disease, it would be towards between 0 and 20 of the x-axis. So stages throughout those. And the last thing is sensitivity and specificity, so at accuGFR 60. So if we can just show you the final metrics here. So basically at P20, so a little bit squeezing down the regions a little bit more, not P30, but P20, so plus or minus 20% of mGFR values. We see an improvement with accuGFR of 78% versus 65% by CKD-EPI. The staging concordance, so looking at the different stages, 68% versus 58% by CKD-EPI. And then finally, giving a little bit back on sensitivity, however, increasing specificity from 0.66 to 0.83. In addition, as I mentioned before, we do see a 37% reduction in GFR misstaging and a 27% reduction in the number of false positives and false negatives at that 60 GFR threshold. So the last slide I wanted to talk about here before we give a summary is some applications that we've seen that might be relevant to using accuGFR in pharma. The first being patient selection, so better patient selection during enrollment. We do have some more examples of how this was used by some of our clients that we can share if there is interest. Basically, better selection of patients during enrollment so that if you want to have a patient between stages 2 and 3, you're more confident that those patients are in stage 2 and 3 and not some in stage 1 and some in stage 5 using accuGFR. The second is assessment of drug efficacy, so giving you a shorter time to an event or a decision, for example, in a kidney disease trial. So these trials obviously take a long time, cost a lot of money. if you can better select your patients and better give an assessment of drug efficacy with accuGFR versus a simple eGFR, then that would save you time and money. The third is drugs with an acute effect on creatinine. So we have found with some of our clients that certain compounds, certain drugs that they're using for various therapeutic purposes actually do have an indirect effect on creatinine. Thus, eGFR creatinine-based tests cannot be used for their purposes. So this is an alternative to those creatinine-based eGFR methods. And the final is assessment of drug safety. So fine monitoring of kidney dysfunction or nephrotoxicity is another example of an application. The final slide here, again, just a summary. So accuGFR, it's not meant to surpass mGFR. It's meant to be better than eGFR, and thus giving clients more time to a decision, more accuracy, more precision, basically saving them time and money. It is a true confirmatory alternative test. It is presently the only test to outperform the creatinine-based test, which is unprecedented. The creatinine-based eGFR, CKD, EPI, and eGFR MDRD have been around for a long time. They're very valuable. However, we feel that with accuGFR that we have developed a product that is more precise and more accurate, and we have the data to back that up. Final thing to note, again, this was done in development, in the development validation process with the top KOLs in the world, with those KOLs understanding the limitations of the presently used eGFR, CKD, EPI, and eGFR MDRD to help develop the equation for accuGFR.
Thank you very much. We do have several questions that have come in. We have a couple related to urine and the kidney. The first question, is there a mechanistic link between metabolites and the kidney?
Yes, so this is a question I touched on a little bit. So is there a mechanistic link between the specific metabolites on accuGFR and the kidney? So that's a common question that is asked. The answer is no. So, and really that's a good answer because creatinine is not related mechanistically to the kidneys either. So a little bit of background on the kidneys. So the kidneys are our main organs to filter our blood and excrete metabolites into the urine. When the kidney function fails, maybe as a comorbidity with different diseases, maybe a simple CKD, it becomes more porous in certain areas and it becomes clogged in certain areas. So certain things go through the kidney faster, certain things get held up from the kidney. Creatinine was identified many, many years ago, decades ago, as a metabolite, which is a good marker of kidney disease. Now, over the years, many different detection methods, hardware, software developed at Metabolon has enabled us to identify markers, additional metabolites that are actually better predictors than creatinine. I showed you some data how some of those predictors, the four metabolites actually correlate better with mGFR than creatinine or urea. So the importance of having a mechanistic link of these four metabolites to the kidney is really not important. It's just, using the many large cohorts, many different diverse cohorts, we were able to see that consistently these four metabolites are the best in predicting and giving a more precise and accurate GFR readout.
All right, and a follow-up question from a different attendee. Have you performed this analysis on urine samples, and if so, is it validated?
We have done a number of other tests in other matrices, such as urine. So it is not validated. We do not have a method or an algorithm validated in urine as of yet. However, research, our R&D for this accuGFR test is ongoing. We are still monitoring additional cohorts. We are doing stability testing, batch testing, a number of other tests, including in serum, plasma, and in urine, to further the reach of this test in targeted analysis.
All right, perfect. We got a question fairly early on during Robin Parham's presentation. Karen asks, what do you mean by a large number of samples? Could you clarify?
Yeah, so I mentioned that we ran a study with over 12,000 samples. Really, that's dependent on the assay and the run length. Typically, we keep our run lengths less than 5 minutes if possible in our group. So, I mean, the key here is that we're flexible and we just need to have a conversation, really.
Okay, perfect. Sounds pretty straightforward. Another question about the samples. How do they need to be delivered for the targeted assays?
So extractions are dependent on which assay you're running as far as which focus panel or if it's for accuGFR or anything else. However, and also the volume is also dependent on that as well. But for all assays, we just need fresh, naive, frozen at -80, shipped on dry ice. So pretty straightforward, but again, the volumes would be dependent on which assay you have chosen to run.
All right, that makes sense. Another question, what is the turnaround time for accuGFR?
So accuGFR is a very industrialized process. It doesn't take long to run the samples. It would depend on the queue at the time. We do have capacity presently to run 3,000 samples per month. So it would depend on the number of samples you have and the queue that we have at the time of sample receipt. Typically shouldn't take more than a couple weeks and has often been faster than that.
Okay. And what type of equipment are you using for the targeted analysis?
Yep, so in the targeted analysis group, we use mostly SciEx triple quads. We have one 4,000, several 5,500s, we have a 6,500, and we even have the new latest and greatest 6,500 plus. Those are all paired with Agilent 1290 systems. We also have one GCMS, a thermal quantum system that we use for our FAME analysis.
Ah, okay. So quite a full retinue there of capabilities.
Yeah, absolutely.
It appears that we've covered all of the questions. Thank you so much again to Robin Parham, Richard Robinson, and Dr. Brian Kepler from Metabolon for your presentations today. Thank you all so much for coming, and we hope to see you at future CHI Global Web Symposia. Thank you, and have a great day.